Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And so far what we have discussed about the different types of antigen antibody interactions, where we have discussed about the acrylation reactions, we have discussed about the radial immunoassays, we have discussed about the precipitation reactions, and in the previous lecture we have also discussed about the immunoassays. So, within the immunoassay we have discussed about the direct ELISA as well as the indirect ELISA. So, now subsequent to that we are also going to discuss about the sandwich ELISA. Sandwich ELISA is actually a different types of setup. This setup is used to measure the level of antigen such as the insulin in the serum, and in the direct ELISA setup a known amount of antibody is specific antibody to capture the antigen. The antigen is then recognized by the secondary antibody linked to the enzyme. A colorimetric substrate is used to measure the level of antigen. So, in the sandwich ELISA you use the two antibodies, one is the capture antibody. So, what you can do is first you take the well and then you coat this well with a capturing antibody, and then you add your antigen of interest. For example, in this case it is insulin. So, you add the your patient sample, and then the insulin or the antigen is going to bind to this capture antibody. Then you do a washing step, and then you add the additional primary antibody. So, that actually is going to recognize the antigen again, and then you add the secondary antibody which is actually going to recognize this antibody instead of this antibody, and then you are going to add the substrate, and that actually is going to give you the product which is going to be colored in nature, and that is how you can be able to measure the level of antigen present in the patient sample or the biological fluid. So, the purpose of sandwich ELISA is to measure the antigen, whereas the purpose of indirect ELISA is to measure the level of antibody present in the blood. So, in this method a capture antibody is used to collect the antigen from the sample. Afterwards, a second antibody is used to detect the antigen bound to the capture antibody. The second antibody is directed against the antigen using a unique distinct epitope. The antibody is linked to the biotin, and that can be recognized by the avidin or streptavidin HRP complex. In the last step, the peroxidase substrate is used to get a readouts. So, when you immobilize the capture antibodies, the antigen goes and bind to the capture antibody, then you use the detection antibody which is actually going to use the different epitope present onto the same antigen, and then you have the option of either using the secondary antibody or you can use the biotin avidin kind of recognition system, and then you are going to add the substrate, and that actually substrate is going to be processed by the HRP. HRP means horseradish peroxidase, and that actually is going to give you the colored product, and that colored product can be recognized by the colorimetric assays. So, let us see how to do that and what are the materials are required. So, first is you require a capture antibody. So, in this case we are showing you the ELISA for the TNF alpha from the patient sample. So, you are using the anti-mouse TNF alpha monoclonal antibody. It is 1:250 dilution is used for coating into the coating buffer using the ELISA plate. Then you require a detection antibody. So, biotinylated anti-mouse TNF alpha monoclonal antibody is used, and the recommended 1:500 dilution is required in the reaction buffer for detecting the TNF alpha in the sample. Then you require the enzyme substrate. So, streptavidin horse peroxidase conjugate is being used as a detection system, and then you require the standard. So, in the case of this you actually need the mouse TNF alpha standard, so that you can be able to perform the standard reaction, so that you will be able to measure the level of TNF alpha into the patient samples. And the enzyme treatment HRP conjugate. So, this is the substrate what you require to use, and a recommended dilution is 1:250 in a to detect the TNF alpha in the sample. Apart from that, you require a coating buffer. So, this is the composition of the coating buffer. The coating buffer has to be prepared fresh and you have to use that in the span of 7 days. Then you require the assay diluent. So, assay diluent is a PBS which actually can be used to prepare the different dilutions of samples. Then you require the wash buffer. So, wash buffer contains the PBS plus 2 in 20, and then you require the substrate solution which is the TMB and the hydrogen peroxide, and then once the reaction is over, then you require the stop solution which contains the H3PO4 or the sulfuric acid. Then you require the 96 well plate for doing the ELISA, and then you require the microplate reader which actually can be able to capture the absorbance at 450 nanometer, and then you require the micropipette tubes and the plate or the parafilm. The step 1, in the step 1 you have to collect the specimen or the sample from the patient or the biological fluid. So, in the step 1 you have to do a specimen collection and the handling. The specimen should be clear, non-hemolyzed and non-lipidemic. In the case of cell culture, remove any particulate matter by centrifugation and assay immediately or store the sample at -20 degrees Celsius. Avoid repeated freeze thawing cycle. So, even if you preserve the sample at -20, you should not repeat the freeze thawing cycle, because as many times you do the freeze thawing cycle, it is actually going to damage the antigens what is present inside the sample, and that actually is going to reduce its cross reactivity. Whereas, in the case of patient blood, use a serum separating tube and allow the sample to clot for 30 minutes and then centrifuge for 10 minutes. So, in the case if you are processing with the patient blood, you do not need the blood component, you only require the serum component or the fluid component. So, what you do is you just allow the blood to clot and then you take the clear supernatant for detecting the antigen, either it is TNF alpha or insulin or any other antigens. You remove the serum and assay immediately, or you store them at -20 degree. You avoid the repeated freeze thaw cycles. So, that is very important that you should not do the repeated freeze thaw cycle, because that actually is going to reduce the reactivity of the antigen against the antibody. In step 2, you have to prepare the TNF alpha standard dilutions. So, what you have to do is you start with the 1000 picogram per ml and then you do the serial dilutions like as we discussed for the indirect ELISA as well, and ultimately you are going to get the 15.56 picogram per ml dilutions. So, this is exactly what you have to do. You have to start with the stock solutions, and then first you prepare the 1000 picogram per ml, you take the 300 microliter, you dilute it, and that is how you actually are diluting. Every time you are diluting half into the individual ependophs, and that is how you are going to prepare the complete dilutions. So, you take the whatever the content comes along with the kit into a deionized water to get a stock of 30 nanograms per ml, and then you can make the dilutions like this and you vertex the mixture, so that it is going to be a homogeneous dilutions, and then only you are actually going to prepare the serial dilutions, because once you add the things you have to mix this very thoroughly, so that it is actually going to be homogeneous, and then only you take out an additional 300 microliter to make the additional dilutions. The step 3, you have to do the coating. So, add the 100 microliter diluted capturing antibody to each well, and then you incubate overnight at 4 degree and aspirate and wash 3 times with the wash buffer. So, now you have coated your coating antibodies, then you do the blocking. So, exactly the same way you have to do the blocking, so that there will be no non-specific interaction of the antigen to the plastic of that particular well, and then you add the 100 microliter of standard TNF alpha or sample and incubate it for 2 hours at room temperature. Then in the step 5, you aspirate the sample and wash the plate 5 times with the washing buffer, and then step 5 you have to do a detection. So, add the 100 microliter of the detectors to each well. So, detector is a streptavidin HRP conjugate. Incubate for 1 hour at room temperature, aspirate the detector solution, wash it, and then you incubate it with the substrate solutions, and that actually is going to give you the reactions after the some time, and that actually can be read out with the help of the spectrophotometer. Now, determination of the TNF alpha level. So, we are just giving you an example of TNF alpha, but that can be done with any antigens. The mean absorbance of the each set of the sample and subtract the background absorbance from the each mean. Draw a calibration by plotting the standard TNF alpha concentration against the absorbance. So, you actually you plot the absorbance on the y axis and you plot the TNF alpha concentration on the x axis, and that actually is going to give you a curve, and use a regression analysis and you can be able to draw the equation of this particular curve, and then use this equation to determine the TNF alpha concentration into the unknown samples. So, this is all about the sandwich ELISA to determine the level of antigen into the patient sample or the biological fluid. Now, this is the competitive ELISA. So, competitive ELISA is actually based on the competition binding for the primary antibody between the target antigen in a sample and the same antigen that is coated onto the multi-well plate. In this case, what you are doing, the primary antibody is first added to the sample to form the antigen antibody complexes. The sample is then added to the well that are coated with the target antigen. Only the unbound primary antibody in the sample can bind to the antigen coated in the well. Hence, the more antigen in the sample, the less antibody is available to bind the antigen in the well, resulting in a signal reduction, which means what this mean is that you make the antigen antibody antigen complexes into the sample. So, what you do is you take the sample, you add the antibody. So, what will happen is the antibody will go and bind to the antigen. So, as I said, antigen and antibodies are always making a complex in the equimolar ratio. So, suppose you added the 10 microgram of antibody. So, what will happen? The 10 microgram of antibody is actually going to form the complex with the 10 microgram of antigen. So, now imagine that you have the more amount of antibody. So, suppose I added the 100 microgram of antibody and the level of antigen present in your sample is only the 20 microgram. So, what will happen is the rest 80 microgram antibodies are still available. So, they will go and bind to the antigen what is available onto the plate, and that is how they are actually going to give you the readouts. But if you have more amount of antigen, for example, if you have 80 microgram of antigen, then you only have the 20 antibody molecules available, which means as the amount of antigen in the biological fluid is available, it is actually going to keep reducing the available antibody for making an interaction with the antigen which is present onto the plate. This means there will be a competition of antigen between the antigen which is present onto the plate and the antigen what is present onto the sample, and that is why this is called as the competitive ELISA. Either direct or the indirect detection can be used in the competitive ELISA, which means you have an antigen which is actually being coated onto the plate and you have an antibody which is bound to the antigen which is in the sample, and then when you add them to this, so antibody plus antigen is, so antigen star is making a complex like antibody antigen star complex. So, when you have the antigen which is coated onto the plate, this antigen is actually less. So, you have the excess antibody, and that excess antibody is going to react with this antigen, and that is how this antibody has a enzyme. So, that actually is going to process the substrate and will give you the color. The advantage of this assay is that it is highly sensitive, it can be used with the crude sample, and you can be able to use the signal whatever you get can be quantified either by with the serial dilution standard curve. So, you can actually use the different amount of antigen into the solution, and that actually will allow you to measure the level of antigen. The disadvantage is that you require a primary antibody which is very specific for at that particular antigen. It should not have the cross reactive species. So, this is all about the antigen antibody interactions. We discuss about the agglutinations, we discuss about the precipitations, we discuss about the radioimmunoassays, and we discuss about the immunoprecipitations. Within the immunoassay also we discuss about the ELISA, and now we are going to discuss about the Western blotting. So, Western blotting is a technique to detect the proteins what is being blotted onto the nitrocellulose membrane. So, the Western blotting is a popular technique to detect the specific protein present in the crude lysate or the homogenate. It uses the separation of different proteins in the gel electrophoresis, preferably SDS page, and then on the transfer of the proteins onto a solid support such as the nitrocellulose membrane, a primary antibody directed against the protein of the interest, and then secondary antibody is used to detect the primary antibody and give either the color or the chemiluminescent substrate. So, what you have is you have the proteins present on the SDS that you transfer onto the nitrocellulose membrane, then you add the primary antibodies, the primary antibody will go and bind to their target antigens, then you remove the steps with the washing. So, the washing will remove the non-specifically bound antibodies, then you are going to add the secondary antibodies, and that actually is going to bind to the primary antibody, and then ultimately you can be able to add the colorimetric substrate or the chemiluminescent substrate, and that actually is going to give you the signal next to the protein where the primary antibody is bound. So, let us see how to perform the Western blotting. So, the material what you require is the cells, in this case the E. coli which is overexpressing the GFP. You require the protein standards, transfer buffers, transfer membrane like the nitrocellulose membranes, the plastic tray, spatula, blotting sheets, semi-dry electroblotting units that is required to electroblot the proteins from the gel to the nitrocellulose membrane. Then you require the reagent for performing the electrophoresis. You require the primary antibodies to detect the GFP into the proteins. You require the secondary antibodies which is coupled with the HRP, and then you require the developing reagents. In the step 1 you have to prepare the samples. So, the preparation of the sample depends on the cell type. For a tissue, first the solid tissue such as the tumor or the whole liver or the brain, it is first mechanically broken down into the individual cells using a blender and then homogenize it for the sonications. Once the individual cells are obtained, it will be processed as described below. For the cell, individual cells are incubated with the lysis buffer containing detergent along with the protease as well as the phosphate inhibitor cocktail to protect the sample from the degradation. So, the phosphate inhibitor cocktail only you will use if you are interested to probe the antibodies or probe the antigen which is phosphorylated. So, because you do not want the phosphatase to remove the phosphate from the phosphorylated proteins. The step 2, you have to electrophorese the sample. So, your sample was resolved on the SDS page. Then the step 3, you transfer the protein gel onto the blotting membrane, and then before you do the transferring of the blotting membrane, you have the multiple steps like you have to prepare the transfer membrane. So, you have to cut the membrane into the same size as the gel, like if you have a gel of this size, you have to cut the membrane of this size, and then you have to process the membrane in different way depending on which membrane you are using. So, for example, if you are using the nitrocellulose membrane, place the membrane in the transfer buffer and observe that the liquid has wicked the membrane. Areas appeared as a white spot needs a special consideration, which means if you add the transfer buffer, this membrane is going to be completely wet. There will be no white spot present on this membrane, which means this membrane will not be visible. It is actually going to submerge into the transfer buffer and it will take up the same color as your transfer buffer. That will indicate that the membrane has taken up the transfer buffer. If you are using the PVDF membrane, which is actually a hydrophobic membrane, then what you have to do is you immerse the membrane into 100% methanol, because then you have to do a charging step, because the PVDF membrane has to be, because it is a hydrophobic membrane, so it will not bind the protein very nicely. So, it has to be charged with a polar solvent. So, what you have to do is you first incubate the PVDF membrane into 100% methanol for some time, and then you add the transfer buffer, and when you add the transfer buffer should take up the PVDF the buffer, and it should be also does not can show any white color. Decant the methanol and submerge the membrane into a transfer buffer and additional 10 to 30 minutes. So, with this the transfer membrane is going to be ready, and then you have to set up the electroplotting. So, assembly of the transfer cassettes. So, what you have to do is remove the stacking gel from the page and incubate the gel in a transfer buffer to 10 to 30 minutes. To place the pair of blotting sheets onto the anode plates. So, a transfer apparatus is having a anode on one side and the cathode on one side, and then you have to set up the things like this. You have to first put the filter papers, then you have to keep your nitrocellulose membrane, or then you keep your gel, and then you keep your transfer membrane. Or you can do reverse, that you first put the filter paper, then you put the gel, then you put the membrane, and then you keep the next layer of the filter papers. You have to ensure that there is no bubble present between the gel and the nitrocellulose membrane, because if there is a there will be an air bubble present, that area is not going to be will not going to allow the transfer of protein from the gel to the nitrocellulose membrane. So, place the transfer membrane onto the top of blotting sheets and remove the trapped air by rolling the test tube or the glass rod. Then you place the page gel on top of membrane and gently remove the trapped air bubble by rolling the test tube. You place the another blotting sheets onto the top, and then you remove the trapped air bubble by rolling the test tube or the glass rod, and finally keep the cathode plate, usually the red colored cathode plate, and tight the transfer cassette by the 4 screws. So, once you have done that, you have the 4 screw on all the sides, and that actually you are going to put to screw the this particular gel cassette, and then you connect it to the to the power card, and that actually will allow the transferring of the protein from the gel to the nitrocellulose membrane. Then you transfer the protein from the gel to the membrane, place the transfer cassette in the tank filled with the transfer buffer, connect the transfer apparatus to the power supply, and apply the constant voltage. After the transfer, disassemble the whole cassette and carefully remove the transfer membrane and check the transfer by the Ponshu stain. Use a pencil and label the different lane, which means if you got these lanes and this membrane, you have to write the lane number, so that lane number can be used subsequently when you are doing the chemiluminescent development, or you are using the autoradiogrammy or any other method. This number will actually allow you to identify the particular lane. In the step 4, you have to do the blocking is exactly the same, and then step 5, you have to do the probing. So, in the Western blot, the probing can be done in 2 ways. In the 2-step probing, you have to first add the primary antibody followed by the secondary antibodies. So, you have to take the appropriate dilution of the primary antibody, you have to add the primary antibody followed by the washing, and then you have to add the secondary antibodies. Or in a 1-step probing, what you have to do in 1 step itself, you add the primary antibody which is containing the enzyme, which means the primary antibody which is labeled with the enzyme or the fluorescent probe, and that can be used. 1-step probing is very uncommon, because the 1-step probing does not allow you to do the because the sensitivity level is very low with the 1-step probing. Then you have to do the blot development. There are multiple ways to develop the blot which is present onto the membrane. You have the HRP based chromogenic substrates like the 4-chloronaphthol, DAB or TMB, or you can have the alkaline phosphatase enzyme also. So, for that you can use the BCIP NBT system, or you have the luminescent substrates like for HRP you have the luminol and H2O2, or for alkaline phosphatase you have the substituted 1,2-dioxane phosphate. So, all these substrates are allowing you to detect the proteins what is being blotted onto the nitrocellulose membrane. Either you have the colorimetric methods, or you have the chemiluminescent methods. So, both the methods are actually allowing you to detect the proteins. The colorimetric detection has the lower sensitivity compared to the chemiluminescent detections. Uh, So, this is about the Western blotting. So, we have discussed all these steps, and then we have also discussed about the how to perform these steps. And now, to show you how to do these experiments, I would like to take you to my lab, and where the students are going to perform these experiments, where they are actually going to discuss each and every steps how to perform, and what are the different steps you should take, how to remove the trapped air from the air bubbles or trapped air between the gel and as well as the sandwich, how to process the membrane so that it is actually going to be useful for transferring of the proteins from the SDS page to the nitrocellulose membrane or the PVDF membrane. And with these steps you will be able to understand the how to perform the Western blotting in your laboratories.
In this video, we will demonstrate you how to do a Western blot and how to analyze the result using ECL, electrochemiluminescent substrate. So here what we will do, we have to run gel first, then we will transfer. The transfer method, how to do the transfer, we will show in this video. In previous video we have already shown that how to prepare a SDS page gel and how to run protein samples. So in this video particularly we are interested in factors associated with the Western blotting. For doing Western blot, we need membrane and transfer buffer and the transfer medium. This one is we use to transfer this gel to membrane. So here membrane can be two kind. One is nitrocellulose, which has low protein binding efficiency and hydrophilic in nature. Another membrane is PVDF. This is hydrophobic membrane and higher protein binding capacity. So the processing for Western blot is different for nitrocellulose and PVDF. If you are using PVDF membrane, we have to cut the part. Either if you have ready-made pre-cut blots, then no need. If you are taking from a bundle, you have to cut precisely how many wells you want. So after that you have to label front on the blot, where that front side can be used for transferring the protein, and that can be used in previous step, further steps also, like antibody incubation. So here for if you want to use PVDF membrane, you have to charge with the methanol. So since the PVDF is a hydrophobic membrane, you cannot directly transfer in the aqueous medium. First you have to keep in methanol for at least 20 minutes. So after this can be called as charging. So after this we will use that for transfer. So this is pre-soaked in methanol and equilibrated in transfer buffer. So here while doing transfer, we need to consider few things. The buffer always should be in chilled condition. Otherwise, during this transfer at high voltage, it will generate high temperatures. So that may degrade your protein or decrease the efficiency of the transfer. That's why we need to keep the buffer always in chilled condition. And let's start the procedure. So we need a pre-run gel. So we already finished the gel running. In addition to that, we also need sponges which will give cushion to the gel, so that gel may not destroyed during the transfer. So this is this is the cassette we'll use for the transfer. So this is negative side of cassette and this is the positive side. So we are going to keep gel on negative side and positive side the blood membrane. So when we apply voltage from this side to this side, the negative protein, it will be transferred, it will be moved to positive side and it will be captured in the membrane. So first, for doing that, these sponges we need to keep, and also this maybe give some non-specific binding to membrane. So what we will do, we will put blotting sheets on top of this. So after this, you have to remove air bubbles if any present. So once you inserted the blotting sheet, then you have to keep your gel. So here we have to remember that gel, after finishing the SDS phase running, you have to keep in transfer buffer, so that it will give identical condition or equilibration kind of thing during transfer, so that protein transfer may be easy. So this is the gel and keeping on the negative side. So after that, we have to overlay with the membrane. Next, we have to remove any air bubbles if present. We have to overlay with another blotting sheet and remove the air bubbles. Each and every time when you introduce something, you have to remove air bubbles. So this is the final sheet. So this is the positive side of the cassette. Just have to keep like this. These are the screws. We have to tighten it up, then only the contact between the gel and the membrane will be sufficient to get transferred. First, you don't tight initially, you just keep, and after that press the positive side of the cassette, then tight the screws. So all these things should be done in the transfer buffer only unless specified. So this is the chilled transfer buffer. Now we are going to do transfer. Pour sufficient buffer. Keep this ice pack also. If the chilling is not sufficient, then there may be heat generation. So in order to prevent that, we will use this ice pack. So this will keep the buffer cool till the transfer, end of the transfer. So once that is over, you directly take out the cassette and keep. If there is a buffer insufficiency, you can add on top of that. Make sure that the cassette completely submerged, so that the transfer will be proper and there is no air bubbles. So once the setup is over, now you can transfer. Now transfer is going on. So how much voltage we need to give it depends on transfer to transfer, it varies. Generally in our lab we will give at least 2 hours of transfer at 120 volts, which is sufficient to transfer even low molecular weight proteins also. But from instrument to instrument also it varies. You needed to optimize before doing transfer. After 2 hours we have to remove the blood and incubate with the blocking buffer. So I am going to stop here. Remove the cassette, keep it in a tray. Remove the screws properly. Gently remove the sponges. Take out the blood and keep it in blocking buffer. In this condition we have to keep, if you are keeping it room temperature, it is for 2 hours at least. If you are keeping in 4 degrees Celsius, you can keep overnight. The blocking buffer contains skim milk or BSA along with the Queen 20. The Western blot transfer, it all depends on the efficiency, how precisely you are doing the transfer. For example, you should not use your bare hands while handling the blot or gel. So whatever the proteins present on your fingers, it will transfer into gel or membrane, which will give high background during development of the blot. So always use gloves. Apart from that, while handling the instrument, make sure that there may be possibility of electricity, the shock may happen sometime. So we have to, that time also we need to use gloves. And after finishing the transfer, you have to clean all the apparatus properly and dry it for the next time use. After the blocking of the membrane, we have to remove the membrane and incubate with the primary antibody without washing. The main purpose of the blocking is that it will occupy non-specific sites other than the respective protein, so that when antibody comes, it will bind to that specific protein and gives no noise. So after this, we will incubate with the primary antibody for overnight at 4 degrees Celsius, then wash 3 times, at least 15 minutes each, with the TBST buffer or PBST buffer, and again incubate with the secondary antibody, suitable secondary antibody, for 5 hours at 4 degrees Celsius or 2 hours at 3 hours at room temperature. After that, we need to wash properly at least 3 times. Then we will develop the blood with the electrochemiluminescent substrate. In earlier Western blot, how to do Western blot video, we showed how to transfer the proteins to membrane. So we incubated with the primary antibody following secondary antibody and washed thoroughly. Now here we show how to develop a blot. For developing a blot, we need chemiluminescent substrate. In most of the commercially available kits, luminal is the one of the substrate we used for this purpose. So luminal, in presence of hydrogen peroxide and peroxidase enzyme, which present in the secondary antibody, horse radish peroxidase conjugated secondary antibody. This horse radish peroxidase converts luminal to excited state luminal by deprotonating and oxidizing it. So this product, this excited state product gradually leaves the energy by releasing luminescent photons. That light will be detected using this instrument. So these are the commercially available chemiluminescent substrate solutions. So it is available from a wide range of companies. So we have to mix 1:1 ratio. So we have to take out the blood, drain the buffer whatever present properly, so after that you keep blood in between a plastic paper foils. Then we will take chemiluminescent substrate, so after that you have to slowly press and remove air bubbles. This is the tray we use it for developing the blood. So we have to open the system, properly align the tray, and then shift blood to the tray. Once it is over, you have to just close. Here we have to select application. We want bloods, that is chemiluminescent, and what exposure want. You have two options, manual, auto. In auto two options are there, optimal auto exposure, rapid auto exposure. We will choose optimal auto exposure. So you can enlarge the blood also. Once it is over, you just say so this is the developed blood. So as we can see the bands pattern. So this is how we develop Western blot through electrochemiluminescent substrate. So in this video we demonstrated how to transfer proteins to a blot and what are the precautions need to be taken while doing the Western blot, and also how to develop the blot and what is the laying principle behind the developing the blot. So I hope this will help you to understand the basic outline mechanism of how Western blot works.
So, with this we have discussed the demo of the Western blotting, and in that Western blotting demo the student have discussed many aspects related to the Western blotting. So, with this we have discussed about the antigen antibody interactions, we discuss about the agglutination reactions, we discuss about the precipitation reactions, and then lastly we have also discussed about the different types of immunoassays like ELISA as well as the Western blotting. So, with this I would like to conclude our lecture here. In the subsequent lecture we are going to discuss few more immunoassays related to this particular immunology, and we are also going to see how those tools are helpful in answering the different types of biological problems. So, with this I would like to conclude my lecture here. Thank you.